Hello everyone, hope you're all having a really good day. If you recall, we were discussing the first chapter of Plus 2 biology, right? Sexual reproduction in flowering plants was the name of the chapter that we were discussing. I'm Tanuja and let's continue with the chapter. So we were discussing about the topic pollen grain, right? We discussed about its structure, about its size, right? We also discussed the two wall layers of a pollen grain, the exine and the intine, right? Okay, I if you recall, we were also seen some pictures of a pollen grain in a microscope, right? Now we'll be discussing about the stages of a pollen grain. So usually pollen grain comes in two stages. Okay, when the pollen grain is released from a plant, it can be in two situations. One, it can be in a two-celled stage, and the second situation, it can be in a three-celled stage. So, what is this two-celled stage? What is this three-celled stage? So, if we look into a pollen grain, into the development of a pollen grain usually in the initial stages will be single celled. Okay, it will be just basically a single cell inside the pollen grain, but later on as it develops you can see some vacuoles are formed inside the cell. Okay, so imagine this is the first stage. This is the first stage where a pollen grain is single celled, but later on some vacuoles, some free spaces are formed inside the pollen grain. Okay, these are the vacuoles or the free spaces inside the pollen grain. You also have the nucleus. There's no change in the nucleus, of course. And then later on, as the pollen grain develops, you can see that it slowly divides into a two-celled stage, right? Which are those two cells? The vegetative cell and the generative cell. So there are two cells inside a pollen grain, or a pollen grain can be in a two-celled stage. Which are those two cells? The first cell is a vegetative cell and the second cell is a generative cell. Okay, the vegetative cell is comparatively larger and the generative cell is comparatively small. Okay, you can see this over here in the figure. The vegetative cell is the larger cell with a big nucleus, and the generative cell is comparatively small, of course, with a small nucleus. Okay, if we look into detail, the vegetative cell, as I already said, is bigger, right? Of course, it's bigger. It also has abundant food reserve. Okay, what am I trying to say? A pollen grain has two cells inside, and one of the cells is called the vegetative cell. Okay, the vegetative cell is comparatively bigger, and as it is bigger, it has abundant food reserve inside it. Also, it has an irregularly shaped nucleus. It has an irregularly shaped nucleus. So the nucleus does not have a normal shape. Like we cannot define it as a circle or a pentagon or a hexagon or a square. No, it usually has a shape which is not that regular. Okay, so vegetative cell is comparatively bigger, it has abundant food reserve, and it has an irregular nucleus. When it comes to the generative cell, it is comparatively small. Okay, if you imagine it, it can be seen as floating inside the vegetative cell. So the vegetative cell is a huge cell in which the generative cell kind of floats. Okay, so if the vegetative cell has an irregularly shaped nucleus, the generative cell usually has a spindle shaped. The generative cell as a whole is usually spindle shaped. Okay, so it is small, it floats in the cytoplasm of the vegetative cell, as I already mentioned, and it is usually spindle shaped, like a curve. Okay, like a spindle. Now moving on, what I was trying to say is that in most of the plants, in about 60% of the plants, the pollen grain is released in this two-celled stage. But in some cases, the pollen grain is released in a three-celled stage. What does it mean by a three-celled stage? In some plants, just look at what I'm trying to draw. In some plants, this two-celled stage, the pollen grain from this two-celled stage goes into a three-celled stage. How is that happening? The generative cell, which we were talking about, gets split into two. The vegetative cell remains the same and the generative cells gets split into two. So there will be three cells in total, right? The one vegetative cell and two new cells which are formed, right, from the generative cell. And these two new cells are called the male gametes. Okay, so in some plants it will be released, the pollen grain will be released in the two-celled stage, which is the generative cell and the vegetative cell. I hope that's clear. But in other plants, in about 40% of the plants or 40% of the angiosperms, the whole cell is, or the pollen grain is released in a three-celled stage. One vegetative cell and two male gametes. From where did this male gamete come from? The generative cell got split to form two male gametes. Okay, I hope that's clear. Now, as we talk about pollen grains, we should also discuss about its advantages and disadvantages. I mean, what are the real uses of pollen grain? Or how does it help a plant in any way, in reproduction to be exact? How does it help a plant? Are there any disadvantages to a pollen grain? Let's see. If we talk about the advantages, of course, the first and foremost and the most obvious one would be that it helps in reproduction, right? Without pollen grain, there wouldn't be reproduction. There wouldn't be sexual reproduction, to be precise, right? So if a pollen grain, if we talk about pollen grain's advantages, the first one would be obviously that it helps in sexual reproduction, right? That's quite obvious. And the second one is that it's used as food supplements. Okay, usually pollen grains, as I already said, contains a lot of abundant food materials, right? It is nutritious, so we can use it as food supplements. It is available as tablets and tonics, right? So it can be used as food supplements, and also as as a source of nourishment, or as a source of proteins. You have it in the form of tablets and syrups. It's taken by various athletes and race horses. Okay, so these are the basic advantages of a pollen grain, the main one being sexual reproduction. The next topic is the disadvantages of sexual reproduction. Are there any disadvantages? Of course there is. Then the main one is pollen allergy. You've all heard about pollen allergy, right? Some of us may suffer from it, right? Some pollen grains cause a lot of bronchital problems, right? Asthma, bronchitis, respiratory problems to be intact. You'll have a lot of respiratory problems connected to these pollen grains, like asthma and bronchitis and all that. So this is, of course, a disadvantage for humans, right? It's not a problem for plants, but it's, of course, a problem for humans, right? I mean, getting allergy is not the best thing. So disadvantages, the first one is allergies, and of course, bronchial problems, bronchial afflictions, and then some chronic disorders. If there is a prolonged exposure to some pollen grains, some people may suffer from chronic disorders like asthma, bronchitis, and all that. Okay, an example of a plant is parthenium. Okay, the pollen grains of parthenium seems to have cause a lot of pollen allergy. Now moving on, you have a figure over here, the figure 1.6 from your textbook, which is actually some tablets, right? Some tablets made from bee pollen. Okay, yeah. This is some photos of some pollen products, like some energy shakes, some bee pollen cream. You have stuff like that. Okay, so those are advantages, right? They're economic advantages to be in fact. Moving on, in this figure, you can actually see the real products, right, made from bee pollen. This is the figure 1.6 of your NCERT textbook, and you can see a lot of products like there are energy shakes, right? There are bee pollen cream, there are tablets, right? There are a lot of things that we obtain from pollen grains. Okay, moving on. There is one small topic called pollen viability. What is meant by pollen viability? Pollen viability is basically the ability of a pollen to be able to reproduce. Okay, so how long can a pollen still perform sexual reproduction, or how long can a pollen still be active? Right? That's what is known as pollen viability. How long can a pollen be viable? Is there a limit to it? Of course, there is a limit to it. And this pollen viability differs from species to species. Some pollen may have a longer viability or longer life, as we can say, but some pollen does not. Okay. So what does pollen viability mainly depend on? Pollen viability mainly depends on temperature and humidity. These are the two features on which pollen viability depends on. Okay, which are the two features or two factors that pollen viability depends on? The first one is temperature, and the second one is humidity or moisture or whatever. Okay, so these are the two characters on which pollen viability depends on. So what was pollen viability? It was basically the ability of a pollen grain to still be able to reproduce, right? Okay, in the case of rice and wheat, those are two plants, right? Those two grains actually. In rice and wheat, you can actually see the pollen viability is just 30 minutes. Just after 30 minutes, if the pollen grain try to reproduce, or if the pollen grain try to germinate on the pistil, it will not happen. Okay, the pollen is only viable for 30 minutes. So the pollen, after releasing from the male organ, has to reach the female organ and germinate in 30 minutes. Can you imagine that? So a pollen that is released from a male plant of rice or wheat have to release from the male plant and then go to the female plant and germinate over there within 30 minutes. So there is a small time, right? Only a small time is available for the pollen grain of rice and wheat. But in the case of some families like Rosaceae, Leguminosae, and Solanaceae, these are plant families basically. In Rosaceae, Leguminosae, and Solanaceae, you can have months. The pollen viability will be like few months. Okay, so the pollen can take its own time. After releasing from the male plant, it can take its own time. It can relax, it can go for a trip and then come back and do whatever it wants, and then go germinate on the female plant, right? It has months of pollen viability. So the pollen viability largely depends upon temperature and humidity, but it's also it varies depending upon the species as well, right? Here we have seen some families like Rosaceae, Leguminosae, and Solanaceae. Rosaceae is a family of rose. Leguminosae are the family of pulses basically. And Solanaceae is the family where brinjal and all falls in. Anyways, this is what is known as pollen viability. Connecting to pollen viability, there is also one topic that we should discuss, that is pollen storage. What do you mean by pollen storage? Simply saying, a pollen can't be stored inside a fridge, or we can't like collect it in a box and just store it. It will be destroyed. Okay. So if we want to store a pollen without it getting destroyed or without its ability getting destroyed, then we have to store it in some special condition. What is the condition? The condition is that it should be stored in liquid nitrogen. It should be stored in liquid nitrogen, and there is a specific temperature at which we should store it, and that is minus 196 degrees Celsius. Okay, it's a very cold temperature. So how should we store a pollen grain? We should store it inside liquid nitrogen at minus 196 degrees Celsius. I hope that's clear. Okay, so we have a lot of pollen banks where we have the collection of many pollens from many plants, many species, right? All these pollens are stored in liquid nitrogen at minus 196 degrees Celsius. So I hope that is clear. Okay, don't forget this minus sign. Okay, 196 degrees Celsius is completely a different thing from minus 196 degrees Celsius. So don't forget to put that minus over there. Okay, now moving on. We have the second section, the pistil, megasporangium, and embryo sac. So we've been discussing about the male organ actually, right? We were discussing about we are discussing about sexual reproduction, and before getting into fertilization, we were discussing about the pre-fertilization events. And in the pre-fertilization events, we discussed about the male reproductive organ, its structure, right? We also discussed about how the pollen grain is developed. We discussed about microsporangium. We discussed about porogenesis, right? We discussed all about this. And now it's time to discuss the female reproductive organ, right? The female reproductive organ is also as important as the male reproductive organ. So we should learn about its structure. And let me tell you something. If you have understood the male reproductive organ completely, if you have understood the processes completely, then female reproductive organ is also kind of easy. Because there is a similar term like megasporangium over here. There's a similar term like megasporogenesis over here. It's all kind of similar, and you can compare it and study. So it's kind of easy, okay? So make sure that you have understood the first part really well. Okay, it's only then this part is going to be easy for you. Now moving on, the first part or the first term that we have to study is the gynoecium. What was the first term that we studied over there in the male reproductive system? We studied the term andrecium, right? Androecium, andrecium. Here, the term is gynoecium. What does it mean by gynoecium? I told you andrecium is a group of stamens, right? So stamen is basically the male reproductive organ, and a group of stamen, a collection of stamen together is known as the andrecium, right? Here, the female reproductive organ is pistil or carpel, which we've already discussed in the first section. And a group of pistils or carpels is known as the gynoecium. Okay, so what is gynoecium? It's basically a group of pistils or carpels is known as gynoecium. Okay. So gynoecium is simply saying the female reproductive part of the flower. Okay, pistil, carpel, whatever, same thing. And if a plant has only a single pistil, you call it monocarpellary. And if a plant has more than one pistil, you call it multicarpellary. So how will you remember this? Mono simply means one, right? And multi means multiple, two or more, right? Okay, so and I've also already told you that carpel and pistil can be used simultaneously, right? They're almost same. They're actually the same. They're just two different names of a single thing. Okay, so if you have one pistil, you can call it monocarpellary. You'll never call it monopistillary. That seems a little weird, right? So you can call it monocarpellary. So if a plant has only one pistil, or to be simple, if a plant has only one carpel, you call it monocarpellary. And if a plant has more than two carpels, two or more than two carpels, then you call it multicarpellary. Okay? Now, there are other two terms which you should be familiar with, that is syncarpus and apocarpus. What do you mean by syncarpus and apocarpus? What do you mean by the term syn in general? Syn usually depicts a kind of fusion or something like that, okay? So syncarpus means the carpels are fused. So if it is a multicarpellary condition, the carpels can be free or it can be fused. Okay, I'll draw it for you. I'll make it more clear. I'll draw it for you. Okay, imagine this is a carpel. Okay, imagine this is a carpel. This is a plant and you have 4 carpels over here. Okay, you have 4 carpels and all these carpels are free from each other. I mean, these carpels are not attached to each other. These carpels are free from each other. Okay, these carpels are not attached, they are free. And if they are free, you call them apocarpus. They are not fused, so they are known as apocarpus. Okay, what are they known as? Apocarpus. So what do you mean by apocarpus? It's a condition where the pistils or the carpels are free. So it will be multicarpellary, obviously, and it will be free. So what would be syncarpus? Syncarpus simply means these carpels will be fused. It's as simple as that. So the carpels will be fused. It will be fused to form syncarpus. Okay, to form a syncarpus ovary or carpels, right? So you have the syncarpus condition and you have the apocarpus condition, right? The syncarpus is where the pistils are fused together, and the apocarpus condition is where the pistils or the carpels are free. Okay, so that's some terms related to gynoecium. So you'll just have to know what monocarpellary, what multicarpellary is, and what syncarpus and apocarpus actually means. Okay, I hope that's clear. Now moving on, we have a figure of a syncarpus and an apocarpus condition. So the B figure is the syncarpus ovary. Okay, the ovary is syncarpus here. What do you mean by the ovary is syncarpus? It means that the carpels are simply fused, right? But in this case, you can see each carpels are free from each other, and hence we say this is apocarpus, right? This is a figure from your NCERT textbook. I've not taken it from anywhere else. Okay, it's there in your NCERT textbook, and it's quite clear from this figure, right? It's all fused over here, but in the case of the figure C, it's totally free, right? Now we have to learn about the parts of a pistil. So what do you mean by pistil? Pistil is a female reproductive organ which is otherwise known as carpel. Why am I repeating this again and again and again? I've repeated this almost like 4 or 5 times in the single chapter. Why am I doing it? Because we tend to forget, right? We tend to forget what a carpel was. And we tend to confuse between what a carpel is and a pistil. Right now you may not have any confusions, but as you go further, we'll learn about the parts of a pistil, right? That is what we are going to be learning right now. So you'll have terms over there as well. So some people may tend to think that carpel is some part of a pistil. That's wrong. Carpel and pistil are basically same thing. It's just two names, two synonyms, right? They are synonyms, okay? So pistil and carpel are used simultaneously and they are synonyms. Now we are going to learn the parts of this pistil or parts of a carpel, right? Let's see what it is. The first part is the stigma. The second part is the style, and the third part is the ovary. If you remember, we have discussed this in the first figure that I've shown you. When we were discussing about the structure of a flower, I've shown you this, right? I've shown you stigma, I've shown you style, I've shown you ovary, and I've told you at that time that we were going to discuss this later on, right? So here we are discussing the parts of a pistil. So a pistil is basically divided into 3 parts. And if you just recall, a stamen was also divided into 3 parts, right? A stamen was divided into an anther, a connective, and a filament. Similarly, the pistil is also divided into 3 parts: the stigma, the style, and the ovary. So stigma is basically the upper portion. It actually acts as a helipad or a landing pad for a pollen grain. So pollen grain has to come and land on something, right? A pollen grain will come by air or wind or via insects, and it has to land somewhere, right? Just imagine a helicopter. Helicopter always needs a helipad to land on, right? It cannot go land on the rooftop of houses. It kind of needs helipads, right? So as similarly as a helicopter needs a helipad, a pollen grain needs some landing surface, right? And this landing surface is called the stigma. So what is stigma? Simply, it's a landing platform or a landing surface for who? For pollen grain. So it's a landing platform for pollen grains. Okay, so that's what stigma is. The second part is the style. Okay, so what is style? It is the elongated, slender part of the pistil. What was the elongated, slender stalk or elongated, slender part of stamen called? It was called the filament, right? But here, in the case of the pistil, the elongated, slender part is called the style. Style comes right after stigma. So stigma is the landing platform, and then comes the style, which is an elongated, a slender stalk of pistil. And at the basal portion, you have a bulged part, the most important part. That is the ovary. So what is an ovary? Ovary is the bulged part at the base of a pistil. Okay, it's a bulged basal part of a pistil. So you have three parts: the stigma, the style, and the ovary. These are the three major parts of a pistil. Inside this ovary is a blank space. So what do you usually call a space? Wherever it is, what do you usually refer to as space? You can call it a cavity, right? So you have the ovary over here, and inside the ovary you will have a cavity. And this cavity or the space is called the locule. So the ovarian cavity or the space inside the ovary is simply called the locule. Okay, and inside this locule you have the placenta. What is placenta? You've learned about placentation, haven't you? You have, in morphology, if I'm not wrong, you have learned about different kinds of placentation. If not, I'll explain to you what a placenta is later on when we learn about embryo, sac, and ovule. I'll discuss about placenta, but simply keep in mind that inside the locule you have a placenta. So basically, placenta is simply a figure or simply a part of the plant which actually helps the ovary to connect with the ovule. So there is something called ovule that has to come inside the ovary. Okay, so ovary is not hollow. Ovary has an ovule inside. One or two ovules will be there. Usually in typical cases, you will have one ovule inside an ovary. So if you have an ovary, you will have to have an ovule inside it. And this ovule has to be connected by some means to the ovary, right? And that means is the placenta. So placenta, where is placenta located? Placenta is located inside the ovarian cavity or inside the locule. I hope that's clear. So it's simple, right? I mean, we have learned about three parts of stamen, the anther, connective, and the filament. It's kind of similar. You have a stigma, which is a landing platform, and then you have a style, which is similar to the filament in the stamen. It's a slender stalk. And then you have the ovary, which is the bulged portion. Which was the bulged portion in the stamen? It was anther, right? It was at the top. But over here, it's at the bottom. That's the only difference. Now, the term megasporangia. So we have learned about the term microsporangium before. What was microsporangium? It is a part of the plant that carries a microspores, right? The part of the plant that carries a microspore or microspores is called the microsporangium. So what would be megasporangium? Megasporangium is a part of the plant that carries megaspores. Okay, it's as simple as that. So what is megasporangium? Megasporangium is a part of the plant or the female plant that carries megaspores. Okay, megasporangium has a more common name, and that is ovule. I'll be using the term ovule, and don't confuse between those two. Okay, again, they are simply the same. So megasporangium and ovule is megasporangia is plural and megasporangium is singular. And a common name of the megasporangia is the ovule. So where would an ovule be? Ovule would be inside the ovary, right? So ovule would be inside the ovary and it would be connected to the ovary by the placenta, right? So it arises from the placenta. Would it be wrong if I said it arises from the placenta? It would not, right? Because it's connected to the ovary via the placenta, right? So it's simply arising from the placenta only. And then you have the ovule, right? The ovule, it's a common name of the megasporangium. And as I already told you, in typical cases, you'll have one ovule, but you can have many ovules as well. Okay, you can have one or two or many. And I'll even give you examples. In the case of wheat and mango and paddy, you'll only have one ovule. But in the case of some other plants like papaya, watermelon, and orchids, you'll have many ovules. I'll give you a trick to remember this. You've all had mangoes, right? How many seeds does a mango have? A mango has a single seed. So one point that you should remember is that these ovules later develops into seeds. So you can simply look at this number of seeds and you can determine how many ovules they had before fertilization. So ovules after fertilization develops into seeds. How many seeds are there in a mango? Just one seed, right? So how many ovules would have been there before fertilization? One ovule. It's as simple as that. So same in the case of wheat and other paddy, you'll have one ovule. Okay, in the case of mango as well, you'll have one ovule. But in the case of watermelon and papaya, how many seeds can you see? Can you count them? No, there are numerous seeds, right? So obviously there would have been numerous ovules before fertilization. So they are examples for a plant that contains more than one or many ovules. Okay, so now you know what an megasporangia or ovule is. it's time to study about structure of this megasporangia. So I've already told you what an ovule is, or I've already told you what a megasporangia is, but I've not shown you what the structure is, right? So we'll discuss about the structure of an ovule. So before discussing all these terms, I'll show you the figure because it's much more simple that way. Before discussing this figure, you should know the outline of how an ovary or how an ovule lies inside the ovary. Okay, so I'll draw a picture over here. Imagine this is the ovary. Okay, this is the ovary, and you have the style and stigma above. Let's leave that part. Let's concentrate on the ovary. So imagine you have the ovary over here. And inside the ovary, as I already told you, you have the ovarian cavity. It's just a space inside the ovary, which is otherwise known as the locule. Inside the locule, I've told you that you have placenta. Imagine this is the ovarian cavity or the placenta. Cavity can't be drawn because it's a space. How can you draw a space, right? You can't draw a space actually. So you have a placenta over here inside the locule. Inside the ovary you have the locule, and inside the locule you have the placenta. And from the placenta arises the ovary, right? So the dark colored part that I've drawn here is the placenta. That's the placenta. And now I'll be drawing the ovule. See? The ovule arises from the placenta. Okay, so what have I drawn right here? I've drawn the ovary, right? I've drawn the ovary, and then I've drawn the ovarian cavity or the locule, which can't be actually drawn because it's just a space. And then I've drawn the placenta, right? I've drawn the placenta, and from the placenta arises our ovule, right? So we're basically going to study about this ovule, the structure of this ovule, right? So just look at it and you can already understand one term, which is the funicle. The first term is the funicle. I'll be listing the terms over here at the right side, and let's clear it. Let's discuss each one of the terms and move forward. Okay, so you have the funicle. So what is a funicle? Funicle is basically a stalk, okay, a stalk that connects the placenta and the ovary. So I already told you the ovary is arising from the placenta. So there should be something that connects the ovary and the placenta, right? There should be some connection between the placenta and the ovule. Sorry if I'm confusing between ovary and ovule. Those are completely two different things. I'm talking about the structure of ovule over here. What is funicle? Funicle is simply a stalk. Okay, it's just a simple stalk or a connection between the ovule and the placenta, because the ovule obviously arises from the placenta, right? So there's a simple connection between the ovule and the placenta, and that's known as the funicle. Look at the figure over here. So this is the whole ovule over here. The big picture that you can see over here is the ovule, right? And over here you will have the placenta, right? Over here you will have the placenta, and there should be something that connects the placenta and the ovule, right? There should be some stalk or something like that, right? And that stalk is called the funicle. That stalk is called the funicle, right? I hope that's clear. Now, there is a small junction between this funicle and the ovule. So, the funicle and the placenta is connected. Okay, that's clear. I mean, the ovule and the placenta is connected via the funicle. That's clear. There's a small connection between the funicle and the ovule. That is the stalk and the ovule. And that's known as hilum. What is it known as? It's known as hilum. So the second term that you have over here is the hilum. It's again marked in the figure. So what is funicle? Funicle is the stalk, right? Funicle is the stalk that connects the ovule to the placenta. Okay? And there is a junction between the funicle and the ovule, and that's known as hilum. Okay? I hope both these terms are clear. Now moving on, you have the third part, which is known as an integument. What is it known as? It's known as integument or integuments. Okay, it can be in plural because in many plants you have more than one integument. Okay, what do you mean by integument? It's simply the coat of an ovule. Ovule is not bare. Okay, it has some coat for protection. C-O-A-T, not C-O-U-R-T, not this. Okay, you're not going for trial over here. We are learning about science, so it's obviously not C-U-O-R-T. It's C-O-A-T. The coat of ovule or the protection of ovule or the covering of ovule is simply known as integument. In this figure, you can see two integuments, the outer integument and the inner integument. Okay, it can vary from species to species. In a typical case, you have two integuments, the outer integument and the inner integument. They are basically coverings of an ovule. The other term that you can see over here is the nucellus, right? So inside the covering, inside the ovule basically has two coverings. Typically, an ovule has two coverings, which is known as the outer integument and the inner integument. And inside these integuments are a group of cells, okay? Or there's a mass of cells, which is known as nucellus. So what is nucellus? Nothing but simply a mass of cells. So you have the word cell in the word nucellus, right? When you spell the word nucellus, you have the word cell. You have the word cell over there inside. So it's easy to remember that way, right? So what is nucellus? Basically a mass of cells inside the ovule. Now moving on, you have the last part, the embryo sac. The embryo sac. So embryo sac lies inside the ovule, okay, after the nucellus. So there is the outer integuments and the inner integument, and then you have the nucellus, which is generally a mass of cells, and inside that you have the embryo sac. We'll discuss about the structure of embryo sac and what embryo sac is on the later part, but right now just understand there is an embryo sac within an ovule, okay? So you have, where did we start our journey from? We started our journey from funicle, right? Funicle was the stalk, okay? Let's go back to the statements that you had in the slide before. What is a funicle? A funicle, as all these terms, we've already discussed all these terms, right? We're just revising it right now. Funicle is the stalk that attaches the ovule to placenta. Okay, what is hilum? Hilum is the junction between the ovule and the funicle. It's just a point. It's just a junction. Okay. Then you have the integuments, which are the protective envelopes of an ovule, or simply saying it's a coat. It's a cover of the envelope. And then you have the nucellus. What is nucellus? Nucellus are simply mass of cells within the integuments, right? And then you have the embryo sac, which lies inside the nucellus, or lies in that the innermost part of the ovule, which is known as the embryo sac, or the female gametophyte. Usually there will be one female gametophyte or one embryo sac inside a ovule. Okay, so I hope all these terms are clear. Now we have two more terms left, right? The micropyle and the chalaza. So basically these are two sides of the ovule. So if an ovule is an oval shaped structure, or if it can be of different shapes, but imagine it's an oval shaped structure, there are two sides to an ovule, right? And in these two sides, there's a small part of the ovule where the integuments are not present. So imagine this is the ovule and you have the integuments over here. Okay, imagine these are the integuments or the protective envelopes. So you have the protective envelopes over here. I'm drawing the protective envelopes around an ovule, or the integuments around an ovule. So there is a small part where integuments are not present, or there's a small part where the integuments are absent, where there is no protective envelope, where it is easy for anyone to enter. Okay, so why would it be there? We'll know it. We'll know it in the later stages. But just know that there is a small part where the integuments are absent, and that part is known as the micropyle. It's known as the micropyle. So basically it's a small opening, right? It can be defined as a small opening because there is no integuments over there, there is no protective envelope, and it's easy to enter into it, right? So enter into the ovule via the micropyle. So there is another part of the ovule, or there is another part for the ovule, which is known as the micropyle, where basically there are no integuments, okay? It's basically an opening. It's a small opening. And opposite to this micropyle, you have the basal part of the ovule, which is known as the chalaza. It's known as the chalaza. So you have two more terms which I have now discussed. You have the micropyle and you have the chalaza. Micropyle is the small opening where there are no integuments and it is totally easy to enter. And then you have the chalaza, which is the basal part, just lying opposite to the micropyle, right? I'll show you the figure that you have in your NCERT textbook. Here you can see the micropyle. Right, that's a small opening where integuments are absent, and then you have the chalazal pole, which is the basal part of the ovule. So chalazal pole is actually the basal part of the ovule. So this is the sixth term, that is the micropyle. The sixth term was micropyle, and the seventh term was chalaza. Right, so these are the basic 7 terms that you have to discuss when you learn the structure of an ovule or a megasporangium. Okay, I hope that's clear. So that's all about ovule, or that's all about megasporangium. Right, now moving on to another familiar term, megasporogenesis. You're not familiar with this term, but you are familiar with something similar to this. You're familiar with the term microsporogenesis. What is microsporogenesis? Microsporogenesis was simply the formation of, or the process of formation of, microspores from pollen mother cells through meiosis, right? That's how I taught you. There is a process where microspores are developed from a diploid pollen mother cell via the process of meiosis, right? So that's what microsporogenesis was. Simply, megasporogenesis is something similar to that. Megasporogenesis is process of formation of megaspores. So the name has it, right? The name itself defines the whole process. So it's a process of formation of megaspores from megaspore mother cell. So over there it was PMC or the pollen mother cell, but over here it is the megaspore mother cell, right? So you have the MMC over here, the megaspore mother cell, which is diploid. Right, the megaspore mother cell is obviously a mother cell which haven't undergone meiosis right now, so it's diploid. And this megaspore mother cells undergoes meiosis. Right, what does it undergo? It undergoes meiosis. It undergoes meiosis, and as a result, what do you get? As a result, you get the megaspores. How many megaspores would you get? Of course, it's meiosis, right? A cell, if undergoes meiosis, a cell when undergoes meiosis, it would obviously give 4 cells, right, as a result. So as a result, you get 4 megaspores, which are all haploid, because of course it's reduction division. Meiosis, otherwise known as reduction division, and the ploidy will be divided into its half, right? So if the MMC was diploid in nature, the megaspore is obviously haploid in nature, and you will get 4 megaspores because it is meiosis we are talking about, right? So you'll have 4 megaspores. And if you recall, in the case of microsporogenesis also, you have got 4 microspores, right? But there's a small difference over here. What is the small difference? Over there in the microsporogenesis, all those 4 microspores were active, right? All those 4 microspores developed into a pollen grain later on, right? But over here, only 1 megaspore is functional. The rest 3 degenerates as it develops. Okay, that's one point that you have to remember. Only one megaspore is functional. All the other 3 degenerates as it develops. See, over here you can see an actual picture of how megasporogenesis takes place. So usually in the initial stage, you'll only have one cell, that is the MMC, which is diploid in nature. And as you undergo meiosis, you can see 4 cells, 1, 2, 3, and 4. 4 cells, which is otherwise known as the megaspores, which are haploid in nature. Right, so this is just simply a figure that depicts megasporogenesis. Now, you have the female gametophyte. I've already told you that out of the 4 megaspores, only 1 megaspore is functional, right? What does that 1 megaspore develop into? The microspore, 4 of the microspores, 4 of them were, out of 4 of the microspores, 4 of them were active, and 4 of them developed into a pollen grain later on, or the male gametophyte. Over here, only 1 megaspore is functional, hence only 1 female gametophyte is present, and that female gametophyte is called the embryo sac. So there will be, you'll have 4 megaspores in the beginning, which are all haploid in nature, but out of these 4, all 3 degenerate. Only 1 will be functional, and that 1 will develop into a female gametophyte, which is otherwise known as the embryo sac. You have heard this term before, right? The embryo sac lies inside the ovule. You have learned the structure of ovule just before, right? And inside the ovule, inside the integuments, inside the nucellus, or into the innermost part of the ovule, you had the embryo sac. So how does an embryo sac develop? Embryo sac, or a female gametophyte, simply develops from the 1 megaspore that you got as a result of megasporogenesis, right? As a result of megasporogenesis, did you just get 1 megaspore? No, you got 4 megaspores, but out of those 4, 3 were not functional. You only had 1 functional megaspore, and that 1 functional megaspore developed into a complete embryo sac. Okay, so that's what we are talking about. So you have the female gametophyte or the embryo sac, right? And this embryo sac is developed from a single spore, right? A single megaspore. So when we talk about single, or when we talk about one thing, we usually use the term mono, if I'm not wrong, right? We use the term mono. So hence we can say this is a type of monosporic development. It develops from a single spore, am I right? So we can also call it a type of monosporic development, right? So female gametophyte or embryo sac typically has monosporic development. Why did I use the word typically? Because there are other cases also. But for you, in the syllabus, you only have the term monosporic development. So just keep that in mind. Okay. Now, how does an embryo sac develop? Is embryo sac also single-celled? I mean, I already told you, a spore is just, you'll only have 1 spore. So from 1 spore, an embryo sac is developed. And is this embryo sac also single-celled? Because there's only 1 spore. 1 spore is basically 1 cell. And when 1 cell divides, will you get multiple cell? or will you get just a single cell? That's a question, right? So we have to look into it. We have to look into the structure of an embryo sac. Let's look into the structure of an embryo sac. This is again one of the most important topics of your syllabus. The embryo sac. It is very important. Take notes, draw the figure, and clear any doubts that you have. You can ask me in the comments, or you can Google it, or ask your ChatGPT or anything. Okay, clear doubts. Okay, you have to clear doubts. You have to make sure that you have the idea. Your idea should be concrete, okay, because this is a very important topic. So what is your idea of an embryo sac? Basically, what do you know about an embryo sac? You know two things right now. The embryo sac lies inside the ovule. That's one thing that you know about the embryo sac. What is the second thing that you know about the embryo sac? The embryo sac has a monosporic development, or simply saying it developed from a single functional megaspore. So these are the two things that you know about the embryo sac right now. But after this slide, you'll know a lot more. Okay, so basically, I'll just draw a simple figure over here from which you will be able to understand the whole point. Okay, simply saying there was a single functional megaspore. Okay, just follow, just come with me. This is a simple story. Let's look at the story. There was a single functional megaspore which was haploid. Okay, it underwent mitosis. Okay, what did it underwent? It underwent mitosis. Okay, it underwent mitosis to form a two-nucleate structure. So basically, it's not a complete mitosis. We can't call it a complete mitosis. It's simply a division similar to mitosis, but cell division is not followed. So how will that be? You'll have only one cell. You'll still have only one cell, but you'll have two nucleus. So basically, only the nucleus is dividing. Okay, keep that in mind. In the initial stages of an embryo sac development or a megasporangium development, you'll only have karyokinesis. You'll not have cytokinesis. In other terms, you'll only have nuclear division. You'll not have cellular division. Okay, so there was a single cell or a single megaspore which was functional and it was haploid, and from there your division began. Only nucleus divides. So the cell remains single, or there's only one cell, but there are two nucleus right now. And from this two nucleate stage, it again divides. The nucleus again divides. The cell still remains single. Okay, the cell is still single, but you have four nucleus. You have four nucleus, and from this stage, further division happens. Further division happens, and you will have 8 nucleus. You'll have 8 nucleate stage. So there are 3 stages to an embryo sac development. So in an embryo sac development, you have 3 stages: the 2 nucleate stage, the 4 nucleate stage, and the 8 nucleate stage. And all these divisions are strictly free nuclear. What do you mean by strictly free nuclear? It is not followed by cell division, or cell wall is not formed between the cells, right? There is no cell division. There is only nuclear division. It is free nuclear division, and it is strictly free nuclear. Okay. Now, you'll have a structure like this where you have a big cell and you'll have 8 nucleus inside it, right? This is where cell division takes place. So, over here, cell division or cell wall starts forming. Okay, cell wall starts forming somewhat like this. Okay, cell wall starts forming somewhat like this. This is how cell wall starts to form. Okay, we look at the figure and make it more clear. So right here you'll have a single celled, or you'll have a single functional megaspore, right? You'll have a single functional megaspore which was haploid. From that megaspore you got a 2-celled stage, right? So this is the 2-celled stage where you can simply see 2 nucleus in a single cell, or simply you can also say 2 nucleate stage, because that would be more clear. So you have a 2 nucleate stage. There's only one cell, right? But 2 nucleus. So you can say you have a 2 nucleate stage over here, and then you have the 3 nucleate stage. So this is the 3 nucleate stage. Sorry, the 4 nucleate stage. You have the 4 nucleate stage, right? 2 nucleus divides into the other 2, hence you will have 4 nucleus as a result. And then you have the 8 nucleate stage. The third stage is the 8 nucleate stage. Right? You have the 8 nucleate stage. And all these divisions, the first 3 divisions, are actually strictly free nuclear. You will not have any kind of cell division over there. But later on, you can see cell walls start to form and cell division takes place. How does cell division takes place? Slowly, the upper three nucleus, the three nucleus lying at the chalaza end, okay? Basically, chalaza is the basal portion of an ovule, right? So, but in usual cases, the ovule will be inverted. Even though chalaza is the basal part of the ovule, in most cases, the ovule will be positioned in an inverted form. And hence, in this figure, you have the chalaza end at the top and the micropyle end at the bottom. You will be confused because in the slide before, I just told you the chalaza end is the basal part. But over here, I've written chalaza end at the top. Or over here in your figure in the NCRT, you will have the chalaza end at the top. You will be confused why it is positioned in the top. It is because in most plants, the ovule is usually positioned in an inverted form, or it is inverted. And hence, you will have the chalaza end at the top and the micropyle end at the bottom. Okay, I hope that's clear. So at the chalaza end, you have three cells and they are called antipodals. So there will be three nucleus and these nucleus will be covered by cell walls, and you have three different cells called the antipodals. Okay, you have three different cells at the top called the antipodals. At the bottom, okay, when you look at the bottom part, there also you'll have three cells, okay, 6 plus 6 plus 2. That's how you get a 8 nucleate embryo sac. So you have 6 plus 6 plus 2, okay. So you will have three cells at the top or at the chalaza end, which is known as the antipodals, and at the bottom also you will have three cells. So basically, this is how it forms. Just learn it in this way. You'll have 3 plus 3 plus 2 format, okay. This will be the format of an embryo sac. 3 plus 3 plus 2. The first three represent the antipodals, okay. There are three antipodals at the chalaza end. And the second three represents the egg apparatus. Okay, what does it represent? It represents the egg apparatus. It represents the egg apparatus. Okay, and this egg apparatus contains two things. The first thing is the egg, and the second thing is the synergids. Says there is only one single egg present inside an embryo sac. So egg is just one, but you will have to have three over here, right? So the rest two are the synergids. Okay, so the egg is a really precious female gamete. So the precious female gamete obviously needs bodyguards, right? So usually when we see a princess movie or something, you'll usually have two bodyguards at both the sides of a princess. Similar way, the egg has two bodyguards, and those bodyguards are known as synergids. Okay, so the princess over here, or the male, or the main important character, the main character over here is the egg, right? And that egg has two cells at the right and the left side, and they are known as the synergids. And the egg and the synergids together make up the egg apparatus. Okay, and the other two, we were talking about 3 plus 3 plus 2 format, right? So the rest two is at the center, which is known as the polar nuclei. So there will be a polar nuclei at the center. There will be two polar nuclei at the center. So now let me ask you a question. You have already seen the structure of an embryo sac and how it develops. We've talked about the 3 plus 3 plus 2 format. We have talked about the egg apparatus, right? We've talked about all that. My question to you right now is, how many cells are there in an embryo sac? It's a simple question. How many cells are there in an embryo sac? I've already told you there are 8 nucleuses, which is in 3 plus 3 plus 2 format. But how many cells would be there? Just look at this figure. Just look at this C figure. How many cells do you think there are? Okay. The first answer would be 8. Some would answer 7. Let's see what the right answer is. Okay. At the chalaza end, you have 3 antipodals, right? You have 3 antipodals. So you have 3 cells over there at the chalaza end. Okay, I agree. Then you have the egg apparatus at the micropylar end. And the egg apparatus contains of the egg, of 1 egg and 2 synergids. That makes again 3 cells. Okay, I agree. You have 3 cells over there as well. But I never told you there are polar cells. Did I say a term called polar cells? No, I told you there are 2 polar nuclei. And these 2 polar nuclei lie in a single cell called the central cell. So these 2 polar nuclei are best friends, okay? They won't be separated like ever. So they don't have a cell wall between them. These polar nuclei are really close best friends, and they lie together with each other. They are not ready to separate. Okay, so these two polar nuclei said, we don't need separate, sometimes when you go for a sleepover, some friends may say, we don't need separate bedrooms, we'll sleep in a single bedroom, we'll enjoy in a single bedroom, right? In a similar way, polar nuclei doesn't need two separate cells. They need a single cell, and that single cell is known as the central cell. So both these polar nuclei lie in a single cell called the central cell. So that makes 3 plus 3 plus 1. So this is how many cells that you have. So this is about the cells, okay? And 3 plus 3 plus 2 was about the number of nucleus that you had, okay? So now tell me, how many cells are there in an embryo sac? There are 7 cells. And how many nucleus are there in the embryo sac? There are 8 nucleus, right? So that's what is written over here. The embryo sac is 8 nucleate and 7 celled. You'll have a question like that sometimes. I've seen it in previous year questions. You'll have it like, you'll be given a statement. The embryo sac of an angiosperm is 8 nucleate and 7 celled. And you will be asked to explain. So you have to explain all of this, right? You'll not have to explain the development, but you'll for sure have to draw this figure, the figure C. And you'll have to mark the antipodals, you'll have to mark the polar nuclei, you'll have to mark the central cell, you'll have to mark the egg and 2 bodyguards, 2 synergids, right? And then you can also mark the chilazal end and the micropylar end, okay? In this whole slide, I've explained almost everything except just one term. What is that one term that I haven't discussed about? It is filiform apparatus, right? Filiform apparatus. Where do you find it? Where do you find filiform apparatus in this slide? You find it over here at the bottom, right? At the micropylar end, you find the filiform apparatus near to the synergids. You have filiform apparatus near to the synergids. So what would be the function of filiform apparatus, right? Keep that a question in your mind. We'll see in the following slides, okay? Now, the next topic that we'll be discussing is the pollination, okay? So we were discussing about pre-fertilization events, right? We haven't reached fertilization yet. We were discussing about pre-fertilization events and structures. And in that section, we've discussed about pre-fertilization structures. We have discussed about the female reproductive system and the male reproductive system. That's all you have to know about pre-fertilization structures. And now about pre-fertilization events. There is only one single event that happens before fertilization, and that is pollination. The most important event, the most important pre-fertilization event is known as pollination. Pollination is actually a really simple topic. I mean, we've learned it obviously in the smaller classes, or at least we know about it. It's not something really new to us, right? Pollination is simply the transfer of pollens from the male reproductive organ to the female reproductive organ. To be precise, pollination is the transfer of pollen grains from the anther to the stigma. You know what anther is, right? Anther is a part of the stamen or the part of the male reproductive organ which actually releases pollen grains, right? And you know what stigma is? Stigma is a part of a carpel which acts as a landing platform for the pollen grain, right? So pollination is simply the transfer of pollen grains from anther to stigma. Pollination is actually of 3 types. Okay, pollination is actually of 3 types. The first one being autogamy. So what do you mean by autogamy? You've already heard about 2 types of pollination. You've heard about self-pollination, right? And you've also heard about cross-pollination. If not, don't worry. I'll tell you what it is. Autogamy is otherwise known as self-pollination. What is it called? It's called self-pollination. But the term that you'll have to study is autogamy. But for remembering what autogamy is, you can also use the term self-pollination. So it's nothing but simply the pollination happening inside a single flower. So inside a flower, imagine a bisexual flower. What do you mean by a bisexual flower? A bisexual flower is a flower that has both the stamen and the pistil. So in a bisexual flower, you'll have both female and male reproductive organ. But in the case of a unisexual flower, you'll only have one reproductive organ. It can be either male or female. In the case of a bisexual flower, as I already told you, you'll have 2 reproductive organs, both the reproductive organs, the male organ and the female organ, that is the stamen and the pistil. Okay. So autogamy or self-pollination is the transfer of pollen grains, obviously, from anther to stigma. Okay, till that it's exactly the same definition of pollination, but there's something that you'll have to add with it. Okay, so it is the transfer of pollen grain from anther to stigma of the same flower. So it is a pollination that happens within a single flower, right? So that's the term that you'll have to add over there. So what is autogamy? Autogamy is the transfer of pollen grains from anther to stigma of the same flower. So the important term over here is same flower. That is the term that you'll have to highlight. Okay, so you'll have pollination, but it will be within a single flower. I'll draw it for you if you need me to. Okay, imagine this is a flower. I'm not that good at drawing, but still bear with me. Okay, imagine this is a flower, and you have the pistil in the middle. Okay, you'll have the style, ovary, and stigma, right? And you'll also have, as is the bisexual flower, you'll also have anthers, or simply saying you'll have stamen, right? This is a bisexual flower which has multiple stamens. Okay, so you'll have multiple stamens over here. What I'm drawing over here is the anthers. Okay, I'm drawing anthers right now. So you'll have the stamen and you'll have the pistil. So these anthers obviously would release pollen grains. Okay, these anthers would obviously release pollen grains, right? So you'll have pollen grains released. Pollen grains are yellow in color. Okay, I'll zoom the image if you need me to. Okay, you'll have pollen grains released from anther. If this pollen grains lies on, or falls on the stigma of the same flower and then germinates over there, it's called self-pollination, otherwise known as autogamy. So what did I just say? You will have a single flower. Okay, this is a single flower. This is a flower, and in that flower, as it is bisexual, the flower should be bisexual in order for autogamy to take place. So you'll have a bisexual flower, and in that bisexual flower, you'll have many stamens, right? Many anthers. And from those anthers, pollen grains will be released. And if those pollen grains are transferred from the anther to the stigma of that same flower, then that's known as autogamy. Okay, so you have the figure over here. So I hope that's clear right now. So autogamy is simply self-pollination, and it is the transfer of pollen grains from anther to stigma of the same flower. So imagine if a flower is open. Okay, if a flower is open, will autogamy ever be complete? No, because imagine if this flower is open, there will be pollen grains from other flowers also. Imagine there's a flower over here, and there will be other pollen grain of another species, or from another plant. You will have other pollen grains over here. And if the flower is open, the stigma is exposed, right? And it is open for other pollen grains as well. So then it wouldn't be autogamy. Autogamy is a situation when transfer of pollen grains happen from this anther to stigma of the same flower. So another pollen grain, or pollen grain from another flower, is not allowed. So if the flower is open, autogamy can never be complete. But if the flower is closed, okay, what did I just say? If the flower is closed, okay, if the flower is closed in some manner, okay, if the flower is closed in some manner, will the pollen grain from outside ever be able to enter inside? No, right? So autogamy will be complete. So autogamy is, when will autogamy be complete? Autogamy will be complete in a bisexual closed flower. Do you agree with me? Autogamy is only complete if the flower is bisexual and closed. Otherwise, if autogamy can happen if the flower is bisexual, but if it has to be a complete autogamy or an exclusive autogamy, you need the flower to be closed as well. Okay, you can't afford to have the flower exposed or the stamen exposed to the pollen grains of another plant. Okay, right. So that's what the second point tells you. The second point tells you that complete autogamy is rare in open flowers. So if the flower is open, complete autogamy is not possible, or it is very rare. Okay. Now, in the case of open flowers and closed flowers, let me tell you two terms. You have a term called chasmogamous flowers, and you have a term called cleistogamous flowers. You have chasmogamous and you'll have cleistogamous. Chasmogamous flowers are simply nothing but open flowers, and cleistogamous flowers are nothing but closed flowers. So cleisto starts with CL. Closed starts with CL, right? So you can remember it that way. Cleistogamous flowers also start with the letter Cl, and the word closed also starts with the letters Cl, right? So cleistogamous flowers are closed flowers, or they are the flowers which do not open at all, and chasmogamous flowers are flowers which are open, okay, where the stigma and the anthers are totally exposed. Okay, in some plants you can find both. Okay, in some plants like Viola, Oxalis, Camelina and all that, you can find both cleistogamous flowers and chasmogamous flowers. If you look at this figure, this is a figure from your NCERT textbook, you can see cleistogamous flowers over here. Okay, imagine this is the ground level. This is the ground level, right? You have the cleistogamous flowers, or the closed flowers. It's clear from the picture, right? It's clearly closed. You cannot see anything outside. It's clearly closed. So you'll have closed flowers or cleistogamous flowers which lie underground. Not every case you can see in this case that it is cleistogamous flowers are lying below the ground, right? But it's not same in every case, okay? In some cases, cleistogamous flowers may lie below the ground level, okay? And you can also see the chasmogamous flowers over here. The chasmogamous flowers are otherwise known as open flowers. They are known as open flowers. You can see over here the flower is completely open, right? And it is susceptible to the pollen grains of other plants as well, okay? Now moving on, that was the first type of pollination, the autogamy or the self-pollination. And now you have the second type of pollination, which is known as the geitnogamy. You've already heard about cross-pollination, right? Actually, cross-pollination is further divided into two. Okay, you've only heard about self-pollination and cross-pollination till now, right? And self-pollination, we've already discussed what self-pollination is. We've discussed that it is autogamy. It is similar to autogamy, nothing different. But cross-pollination is an umbrella term for two other type of pollinations, which is geitnogamy and xenogamy. Okay, so cross-pollination is an umbrella term where you have two types of pollination inside it, the geitnogamy and the xenogamy. So let's see what geitnogamy and xenogamy is. Okay, so geitnogamy is a condition where the transfer of pollen grain happens from the anther to stigma of different flower of the same plant. So over here, it's not a complete cross-pollination. So what comes to your mind when you hear about the word cross-pollination? Cross-pollination is transfer of anther to stigma of a flower lying on two different plants, right? That would be there in your mind. But that's kind of wrong, because geitnogamy is a type of cross-pollination where transfer of pollen grain happens from the anther to stigma of a same plant, but the flowers would be different. Okay, let me draw it for you. You'll have a plant like this. Okay, you'll have a plant like this, and you will have a flower over here. Again, I'm repeating, I'm not that good at drawing, but you'll have a flower over here and you'll have a flower over here. So both these flowers are lying on a single plant, right? So if pollen grains are released from the flower at the top, okay, imagine pollen grains are released from the flower at the top, and this pollen grain, obviously it's released from the anther, right? And this pollen grain travels all the way to the bottom to reach the other flower, right? Which is lying on the same plant. So it reaches over here and it falls on the stigma of the second flower. So you have two flowers, flower 1 and flower 2. Right, and the pollen grain is released from flower 1 and it falls on flower 2, right? So this is geitnogamy. So here transfer of pollen grain, of course, happens from anther to stigma of a different flower, but the plant is same over here. So transfer of pollen grain from anther to stigma of a different flower on the same plant or of the same plant is called geitnogamy. So it's not a complete cross-pollination. It's not self-pollination either, because self-pollination would happen when it is happening inside a single flower, right? But here the flowers are different, so it's not completely. We can't call it autogamy. We can't call it xenogamy, or we can't call it a complete cross pollination as well. So it's in middle, right? It's in middle of xenogamy and autogamy. So it's not completely cross-pollination, but by function it is cross-pollination. So by function, what do I mean by function it is cross-pollination? Transfer of pollen grain from anther to stigma is happening between two different flowers. So hence we can say functionally it is cross-pollination. The function that is happening over here is a transfer, and the transfer is happening between two different flowers, right? So we can say it's functionally cross-pollination, but it is happening within the same plant, right? So genetically it is autogamy. Okay, it is autogamy or self-pollination. So we can say that functionally geitonogamy is cross-pollination. So what do you mean by cross-pollination? Transfer of pollen grains between different flowers, right? So here the transfer is happening between different flowers. So functionally it is similar to cross-pollination. But those flowers lie on the same plant, right? So genetically, is there a difference? No. Genetic difference only happens if the transfer happens between two different plants. But here the plant is the same, right? Plant is the same. Hence, we can say that functionally it is cross-pollination, but genetically it is autogamy. Genetically it is similar to autogamy, but functionally it is similar to cross-pollination. Okay, I hope that's clear. Now moving on to the last type of pollination, which is the xenogamy, or it's complete cross-pollination. Here transfer of pollen grains happens from anther to stigma of different plants. The plant itself is different. So you'll have one plant over here. Okay, you'll have a flower. This is the first plant, and then you have another plant over here. Okay, let's draw some more flowers. Okay, you have another plant over here, right? This is a completely different plant. So if the pollen grains is released from the first flower, and if it moves to the second flower and falls on the stigma of the second flower, then this is known as cross-pollination or xenogamy. Okay, so I hope you are clear with both the terms geitonogamy and xenogamy. So geitonogamy is actually an intermediate between autogamy and xenogamy. So these are the main important three types of pollination. Okay, so here you have two figures. This is nothing but a closed or a cleistogamous flower and a chasmogamous flower. Okay, this is a closed flower and this is known as cleistogamous flower, if you recall, and this is an open flower. Of course it's open, right? It's a sunflower, it's open, and it's known as a chasmogamous flower. It's known as chasmogamous flower. It's nothing but closed and open flower. Okay, there's two figures for your reference. Okay, now we have another topic called the agents of pollination. So you already know that what pollination is, and now we have just discussed about kinds of pollination. The next topic, the next subheading we have under the main heading pollination is agents of pollination. So how many agents are there for pollination? Pollination mainly what do you mean by agents of pollination? Let me ask you that before getting into the topic. What do you mean by agents? Agents is basically anything that helps in pollination. Okay, any factor that helps in pollination is known as agents of pollination. So there are different types of agents. So this whole table describes the important agents of pollination. Okay, it's not a table from your NCERT textbook. I have took it from outside. But anyways, pollination mainly has two agents. Okay, two agents. The first one is biotic, and the second one is abiotic. What do you mean by the term biotic? Biotic means living. Okay, biotic means something that is alive, something that is living. And abiotic means something that is non-living. Something that is non-living. So there are two types of agents, abiotic and biotic. The abiotic one would comprise of wind and water, because wind alive? Of course not, right? Wind and water both are not alive, right? They are abiotic factors. And over here in the biotic factors, you have many factors like insects, you have snails, you have insects, you have birds, you have bats, and you have other vertebrate animals as well, right? So in the case of abiotic factors or abiotic agents, you have wind and water. And in the case of biotic agents, or living agents, you have bats, birds, insects, snails, and other vertebrates as well. Okay, now let's discuss the abiotic factors or abiotic agents in detail. The first abiotic agent would be wind. Okay, so the first type of pollination that we're going to discuss in the abiotic section would be the wind pollination. So of course, wind is an abiotic factor, right? Wind does not have life. So wind is an abiotic factor. Before learning all this, just imagine a substance that has to fly with the wind currents. Okay, there are wind currents and something has to fly with it. So it should be lightweight. Obviously, something that is heavy wouldn't easily fly, right? So it should be lightweight. So the pollen grain over here, which is, having wind pollination, the pollen grain of the plant that's having wind pollination should show some conditions or should have some features, right? And the first one would be that it is lightweight. It should not be heavy. It should be lightweight, right? It should be lightweight. That's what's written over here. The pollen grain should be light. And of course, as it passes through the wind, it should not get stick to other things other than the stigma, right? It should reach its destination right on time. Okay, you know that some plants like rice and wheat only have 30 minutes, right? 30 minutes of pollen viability. So it should reach directly. It should reach directly to the stigma without sticking anywhere else. So the pollen grain should be non-sticky as well. Right, the pollen grain should be non-sticky. So the first feature is that it should be lightweight, and the second feature is that it should be non-sticky. Okay, and imagine a plant with a stamen like this. Okay, I'm drawing two kinds of condition over here, and you will have to tell me which is the best for wind pollination. Okay, you'll have a stamen like this, which is kind of covered by the petals. Okay, so these are the petals. Again, I'm not so good at drawing, but just imagine these are the petals. So the stamen is kind of covered over here. So this is a covered condition. The stamens are kind of covered. But over here, you have another plant. Okay, you have another plant which with stamens which are exposed. Okay, the stamens are lying outside to the petals. The stamens are completely exposed, right? See over here, the stamens are exposed, right? So here you can see that in the second figure, the stamens are exposed. In which case do you think wind pollination would be easier? The covered case or the exposed case? Of course, it's common sense that in the exposed case, it would be easy for the wind to pass through the anthers, and it would be easy for the pollen grains to be carried by the wind currents, right? Because the pollen grains would be lying at the anther surface, and as wind passes through the anther, it's easy for the pollen grains to fly along with it. But in the case of the covered plant, the pollen grains will be lying inside the petals. So even if wind goes through it, the pollen grains will not be affected by it, right? The pollen grains will not be affected even if wind goes through it. Okay, so it's always good that the stamen is well exposed. That is the next feature. That's the next feature of the stamen. The stamen should be well exposed. And imagine a stigma. Okay, stigma should be feathery. If the stigma is rigid and if the stigma is hard, the pollen grain that comes through the wind may not easily land on it. But imagine if the stigma is feathery. If the stigma has a lot of feather-like structures, or if the stigma is simply feathery, then the pollen grain would land on the stigma easily. Right, as wind carries the pollen grain, it would be really easy for the stigma to catch it if it is feathery. So that's the next point. The stigma should be large and feathery. So the stamen should be well exposed and the stigma should be large and feathery. I hope that's clear. Okay. Now, usually in the case of wind pollination, there will only be one ovary inside the ovule. One ovule inside the ovary. I always confuse between them. You shouldn't. Okay, you shouldn't confuse between them. Ovary is something that lies outside and ovule lies inside of the ovary. Okay, so in the case of wind pollination, there would be only one ovule inside the ovary. I hope that's clear. An example of a plant which carries wind pollination is simply our corn. We all love corn, right? We all eat sweet corn for snacks, right? So corn is an example which performs wind pollination, okay? And wind pollination is usually common in grasses. So this is the first type of abiotic pollination that we have to study. The second type of abiotic pollination is the water pollination. Before that, let me show you a picture of a plant which performs wind pollination. You can clearly see over here that the anther is exposed and the stigma is feathery. Okay. Now moving on, you have the second type of abiotic pollination, which is the water pollination. The water is water alive? No. Water does not have life, right? So water is also an abiotic factor. So water pollination is the second type of abiotic pollination you are having, and this water pollination is comparatively rare. Okay. In angiosperms, you don't see many angiosperms in water. So hence water pollination is kind of rare. It's only seen in monocotyledons. It's only seen in 30 genus or 30 genera of monocotyledons. It's not that common in dicotyledons. Okay, so it's comparatively rare. And in most cases, water pollination is so common in algae, bryophytes, and pteridophytes. You've already heard about algae, bryophytes, and pteridophytes. If I'm not wrong, you've heard about it in +1. In your third chapter, in plant kingdom, right? You've heard about algae, bryophytes, and pteridophytes. All these have water pollination as a common method for transferring pollen grains. But over here in angiosperms, it's comparatively rare. In this chapter, we are learning about the sexual reproduction of angiosperms, right? So it's comparatively rare. And water pollination can also happen in two types of water: fresh water and marine water, obviously, right? Freshwater plants which perform water pollination, some examples are Vallisneria and Hydrilla. So there are two freshwater plants that you have to keep in mind. Freshwater plants that perform water pollination. The first one is Vallisneria. Vallisneria. And the second one is Hydrilla. You would be familiar with the word Hydrilla, but you will not be familiar with Vallisneria. It's okay. It's a new plant. It's fun to learn new things, right? So Vallisneria and Hydrilla are two freshwater plants that show water pollination. And marine plants that show, there are marine plants as well. There are marine plants as well. And one example of a marine plant is Zoster. Zoster. It's basically a seagrass, okay? It's a marine seagrass. Its name is Zoster, and it also shows water pollination. But don't you misunderstand, okay? All plants lying in water does not show water pollination. All plants that are in water does not show water pollination. You can look here. Water hyacinth and water lily. Those are two different plants. Water lily, of course, it has a beautiful lily flower, right? Water lily and water hyacinth does not show water pollination, even though they lie in water. So water hyacinth and water lily lives in water, but they do not show water pollination. Instead, they show wind pollination, okay? So don't you misunderstand that all plants lying in water show water pollination. No, it doesn't, okay? Some plants show water pollination, yes, but in the case of water hyacinth and water lily and plants like that, you cannot see water pollination. Instead, you can see wind pollination. So this is what water pollination is. So let's look at examples of water pollination. The example of Vallisneria, how water pollination happens in Vallisneria and all, right? Water pollination in Vallisneria is actually kind of interesting. Okay, water pollination is really interesting in Vallisneria. How does it work? In the case of Vallisneria, if this is the water surface, imagine this is the water surface, and you will have the plant inside. But the male flowers, will release pollen grains to the surface of the water. Okay, they will release pollen grains to the surface of the water. So you'll have the male flowers at the top, and they'll release pollen grains to the surface of the water. So this is the surface, and pollen grains will be lying on the surface. So obviously the female flowers should reach the surface in order to receive the pollen grains, right? So the female flowers, of course, does that. The female flowers is grown on a stalk, and it reaches the surface of the water. Okay, it reaches the surface of the water. So even though the plant is lying inside, female flowers will be lying outside in order to carry the pollen grains that released on the surface. Okay, in order to catch the pollen grains that are released on the surface. This way the pollen grains can easily land on the pistil of the female flower. Okay, this is how water pollination happens in Vallisneria. I'll show you real figure. Look at this. The female flower is over here, right at the top on a long stalk, right? You can look over here. This is the female flower. Okay, it has a long stalk, and the top of the female flower, or the stigma of the female flowers, lie at the surface. This female flower lies on a long stalk, and the stigma lies at the surface in order to catch the pollen grain that is released onto the surface of the water. Okay, now moving on, you have water pollination in seagrass, or in the case of seagrasses like Zostera. Okay, I've taught you the water pollination that happens in Vallisneria, which is a freshwater plant. Now I'm discussing the water pollination in seagrass, which is a marine plant. Okay, in the case of water pollination in seagrass, the flower is completely submerged underwater. So if this is the water surface, let me draw it for you. If this is the water surface, the plant is completely submerged under the water. So if imagine this is the plant. This is not how it would look because it's a grass, but still imagine this is a plant. I'm not really good at drawing, I already told you. So imagine this is the plant, okay? So if this is the plant which is submerged completely under the water, and if the pollen grains are released onto the surface, then obviously the pollen grains has to reach under the water. Right, because it's not as in Vallisneria. Unlike Vallisneria, the plant is completely submerged in water over here in seagrasses. So if the pollen grain is released onto the surface, those pollen grains have to get into the water. Right. So the pollen grains should not be easily destroyed by water. It should not decay. Right. For that, these pollen grains will have a special mucilaginous covering. So these pollen grains that have to go under the water and fertilize over there, they'll be having a special mucilaginous covering to prevent wetting and damage. Also, it has to go under the water, right? Hence, it has a long ribbon-like structure. It's not in round shape, it's long and it's ribbon-like. So this is how it would look. Okay, it's long and ribbon-like, and it has a special mucilaginous covering to prevent it from decay. So this is how water pollination happens in seagrasses. Okay, so I've already explained water pollination and wind pollination. And in water pollination, we have learned about water pollination in Vallisneria and in seagrass. So that's all about abiotic factors or abiotic agents of water pollination or abiotic agents of pollination, simply say. Okay. So that's all about abiotic pollination or abiotic agents of wind pollination. Now over here you have a term or a statement. What does the statement say? The statement says that both wind and water pollinated flowers are not very colorful, right? They're not very colorful and they do not produce nectar. Why is it so? Why would you think it is so? Just imagine, why would you need nectar and colorful flowers? Nectar and colorful flowers are made in order to attract insects or other animals or other living biotic agents. So all this nectar and the colorful flowers are made to attract the living or the biotic agents of pollination. But over here we were discussing about abiotic factors. So both wind and water are abiotic factors. They don't have anything to get attracted to. They don't have a life, right? They are not living. So you don't need nectar and colorful flowers in order to attract them, right? It's present in nature. You don't need any kind of nectar or colorful flowers, right? That's why both water pollinated and wind pollinated flowers does not have any kind of nectar or colorful flowers. So that's all about abiotic agents of pollination.